In this session, we are going to dive into what we call sterilization by moist heat. And sterilization by moist heat is done in three ways, whereby we use moist heat, whereby we can use moist heat that is below 100 degrees centigrade, or we can use moist heat at 100 degrees centigrade, and we can use moist heat above 100 degrees centigrade. So these are the three ways we can use moist heat during sterilization. And what is moist heat? You can even call it a steam. And this steam we use saturated steam. So the steam we are using in this session must be saturated. So it is the one we are using, and this steam sterilizes medical equipment, laboratory glasswares, culture media sterilization, culture plates, and surgical usages or PPs which we use for during surgery by coagulation and denaturation of enzymes and structural proteins. So we are seeing all these moist heat, their mechanism of action is by denaturation. They denaturation and coagulation of enzymes and proteins. So it is coagulation of proteins because enzymes are part of proteins. So that's how moist heat acts. And this one is an alternative for the dry heat. So in moist heat, below 100 is known as pasteurization. This one is known as pasteurization. We call it pasteurization because it was developed by Louis Pasteur, whereby the most commonly preserved materials here is milk and the other beverages. So beverages plus milk, beverages can be soda, can be wine, can be beer, plus milk are preserved or are sterilized using pasteurization, which is done in two ways, either at 72 degrees centigrade or at 63 degrees centigrade. When it is done at 72 degrees centigrade, it is known as flash method. So this is flash method, which uses at 72 degrees, whereas 63 is Holder's method. So at 72 degrees, it is done for only 15 to 20 seconds, whereas at 63 is done for 30 minutes. That is what we can call pasteurization of milk and other drinks or beverages, either at 72 for 15 or 20 minutes, that is the flash method, whereas Holder's method is for 30 minutes. And in these two, flash method is preferred during pasteurization. That is sterilization by moist heat below 100 degrees centigrade. Whereas at 100, we have pasteurization by boiling. We can use boiling, whereby we can boil substances at 100 degrees, or we can use steam. And this steam we use can either be single exposure or intermittent, what we call tindalization. So tindalization, that's where you expose intermittent steam at 100 degrees centigrade for whereby every time you expose it at 20 minutes for 3 consecutive days. So here you use 3 consecutive days. That is during steam by intermittent heating, whereby this one is used for heat labile substances like when we are forming Loeffler's serum slope and when we are preparing aerogen media, that is Lawrence-Jensen media, for culturing mycobacteria. We can use tindalization method where we expose serum, egg, and other substances like carbohydrates. So at this point, we use for substances like egg, like serum, like sugars, which are not heat stable above 100. So we can sterilize them by using intermittent exposure. That is 20 minutes at 100 degrees using steam for 3 consecutive days, whereby the first day is for killing vegetative forms, then we keep it, allow it for the spore forms to germinate, then the second day we expose, we again keep to allow the spore forms to germinate, then the third day, by third day we have killed both the vegetative and spore forms of all kinds of microorganisms. So this one can also, with the tindalization, we can maximize and get what we call 100% sterilization, which is different from single exposure, whereby you expose steam to substances at 100 degrees. So the preferred is tindalization. Then lastly is above 100, which is our major topic today. And sterilization by moist heat, that is saturated steam above 100, is what we call steam sterilizer, or what we are going to call autoclave. So this one we are going to call it autoclave. And it is our topic of today, where we are looking at steam sterilizer or autoclaving by an instrument known as autoclave. And as we begin autoclave, if it is your first time on this channel, please, we urge you to subscribe. That is the only payment you can give us, by saying thank you. That is subscribing if you haven't subscribed. Like the video and recommend these videos to many others that we might continue to grow. So with autoclave, this is a lab instrument or a lab equipment used to completely sterilize lab materials, hospital used glasswares and wastes before we expose them off. So we can use autoclave to sterilize curvature media before we prepare, to sterilize used lab glasswares, to sterilize surgical equipment which are used during surgery, and also before we discard biohazard or bio-wastes, we first sterilize them. And pharmacists also use autoclave to sterilize ingredients used in pharmaceutical industries. So this is basically autoclave, and nurses are also using it during surgical procedures to sterilize gauze and other equipments. So with this autoclave, what is autoclave? This autoclave is working at two things. So this is the diagram or the structure of an autoclave. And this diagram of an autoclave, we are seeing a power source. We see this is the heat source, or this one from the electricity. Some autoclaves can use electric or they can use gas. So if you don't have electricity, you can also have a gas autoclave. And this autoclave, it has the water outlet. This is water outlet. After sterilization, you can open to drain the water when it is dirty. Then this is the switch where we can switch on and off, then the timer, and then we have the perforated. This is the perforated stand. Perforated stand where we place our loaded tray. So our loaded tray is placed on this perforated stand within the autoclave after filling with water. Then we are seeing these are the screws. It has the screws which are used to tighten after loading the autoclave before beginning sterilization. Then we have the pressure gauge. This is the pressure gauge. This is the handle. We have the handle. Then this is the exhaust valve, or we can call it air remover. This is air remover, or you can call it exhaust valve. Then this is the safety valve. So this one becomes the safety valve. Then this is the handle. So this is the handle we use when we are removing the cover or when we are placing the lid. So this one you can call it a cover or you can call it a lid. So all this, the pressure gauge, the pressure gauge, the handle, the safety valve, together with the air remover are all found on the lid of the autoclave. So these are the parts of the autoclave. So in an exam, if they ask you to draw and label an autoclave, these are the things you can draw. So these are the stands. So these are the stands where the autoclave is able to firmly be pressed in an environment. And this is sterilization by autoclaving, which we are using temperature above 100. It contains phases, or you can call them stages, whereby phase number 1, you can call it air removal. Some books call it purge phase. So air removal phase or purge phase. Then we have the heating time phase. Heating time. Then we have the holding time. Holding time. Then we have what we call the cooling, or you can call dry it. So these are the 4 phases under sterilization cycle. Air removal, this one is after loading the tray in an autoclave, you have tightly closed with the lid, then you switch on. The first thing is when steam is being generated by the power, the steam displaces the air which is inside the autoclave. And that's why after loading, we open the air remover valve and we leave it for the steam to displace the air present in the autoclave. So this is the first process in sterilization cycle, whereby we remove air, whereby the steam being generated displaces the air inside and passes through what we call the air remover valve. That is the initiation step. Then when all air is removed, we come and close the air remover and heating time begins. Heating time is the time taken for the autoclave to achieve 121 degrees centigrade temperature at a pressure of 15 pounds per square inch. That is the pressure we use. And this one is what we are calling heating time. So after closing the air remover valve, the temperature at the gauge begins to rise and the pressure. And when it reaches at 121 degrees centigrade at 15 pounds per square inch is what we call the heating time. So the time taken for us to achieve this so that sterilization can start is what we are calling heating time. Whereas holding time is the time it takes for the materials loaded in the autoclave to be sterilized. That is what we are calling holding time. And many times at 121, we use average of 15 to 20 degrees, 15 to 20 minutes, depending on the materials being autoclaved. So this is the average, but sometimes it can range from 10 to 40 minutes, depending on the materials being autoclaved. But the most commonly used in the hospital setting is 15 minutes being the holding time. the time taken to completely sterilize equipment in the autoclave when it is at a high temperature of 121 degrees centigrade at 15 pounds per square inch. Then the last phase, after sterilizing, we switch off the autoclave. After switching off the autoclave, we open the air remover and the safety valve, and we allow it to cool to the temperature below 100. And that is what we are calling. So this is the last phase, whereby we open the air valve and we allow the autoclave to cool below 100 degrees centigrade. After cooling, we open the air valve and we allow it to cool again. Then we can remove the lid and we unload our equipments which have been being sterilized. So these are the stages or phases of sterilization cycle. Don't forget they are only four. We have the air removal or the purge phase, we have the heating time phase, the holding time phase, and the cooling phase. So after we have done that, we want to look at the principle of the autoclave. We know that water, because we load the tray with water, and we know that water boils at 100 degrees centigrade, but it boils when this temperature is equal to the atmospheric pressure. So at atmospheric pressure, water boils at 100 degrees centigrade. That is in open system. But when it is in a closed vessel, first of all, the pressure increases. Pressure increases above 100, increases greater than 100 degrees centigrade. At the same time, according to the rules, when pressure increases, also temperature increases. So increase in the pressure leads to increase in the temperature. That is at 121 degrees centigrade. Sometimes if you want for quicker results, you can use even 134 for 3 to 4 minutes. So another time we can use is, you can use 134 degrees centigrade for 3 to 4 minutes. It can also be used, but the most preferred is 121 degrees. So when you are boiling water in an open system whereby the pressure is equal to the atmospheric pressure, water boils at 100. But in a closed vessel, when the pressure increases above 100, that is at 121 degrees. When pressure increases, that is when we increase it to 15 pounds per square inch, also the temperature increases above 100, that is 121. And this increase in the temperature is good enough to cause complete sterilization. It causes complete sterilization. How? By killing both the vegetative forms or microorganisms and non-vegetative, that is spore forming. So this one enables complete sterilization by killing all forms of microorganisms. So autoclaving enables complete sterilization by killing both vegetative and non-vegetative, that is the spore forming organisms, causing the articles to be free of all contaminations. And this is done by sterilization at a pressure of 15 pounds per square inch for 100 at 121 degrees centigrade for the holding time of 15 minutes. When we are using 15 minutes, but we have said the holding time can vary depending on the materials being autoclaved. So this is basically the principle behind autoclave, that water most commonly boils at 100 when the pressure is equal to the atmospheric pressure. That is 770 degrees or 1 atmospheres. But in a closed vessel, the pressure increases above atmospheric pressure, that is at 15 pounds per square inch. Also, when pressure increases, it leads to increase in temperature above 100, that is at 121. So if we load materials and we sterilize at 121 degrees centigrade at pressure of 15 psi for 15 minutes, it is enough exposure time to kill both the vegetative and spore-forming organisms, achieving complete sterilization. That is what we can discuss. Before we begin the procedure, we want also to look at the controls used in sterilization. What could be the controls used? And the controls can be biological, they can be chemical controls. These are the major ones we have. And under biological control, we have a bacteria which is a thermophile, that is Styrothermophilus. Styrothermophilus. That is Bacillus Styrothermophilus. This is a thermophile which can be used as a control because for it being a hyperthermophile organism, it can survive at high temperatures. So we normally use it as a control that when we find when it is already killed, it tells us that we achieved maximum sterilization. And under chemical, we can use autoclave tape, masking tape, or we can use Brown's control. These are the two chemical controls we can use, and also not forgetting the controls like thermocouple. These thermocouple, these are inbuilt, and it helps us to monitor temperature. It is an inbuilt thermometer that monitors the temperature to make sure it is at 121. Then also Brown's control can be used, but the most commonly used control is the autoclave tape. And it is always colorless, but when sterilization is achieved, it turns to black. There are some diagonal strips which are black in color that are formed. That when this is the tape being used, after sterilization is achieved, it forms black diagonal lines. That is the controls we can use, three of them: biological, chemical, and thermocouple, which you can even call it TST. That is temperature, then and time. Temperature, sterilization, and time, TST. Then we have the chemical, that is autoclave tape, which is commonly used. So after looking at the components of the autoclave, looking at the phases, and then the principle and the controls, now we can end our discussion by looking at the procedure. And under the procedure, we begin by identifying the materials to be used, to be autoclaved or sterilized. After identifying the materials, what do we do? After identifying the materials, we load them into the autoclave tray. And this autoclave tray is perforated. It is not a complete glass without aeration. So it is perforated. So what we do, we load our materials to be autoclaved within the tray. That is, if this is, we are going to prepare culture media, this is the bottle containing the culture media. So this culture media in this bottle, we don't fill it to the brim. We have to leave a space to allow pressure to build up. Then the cover is, it must be loosened. So you loosen the cover so that we prevent air from building up, causing fatal problems during opening. Then if we are having also others, we don't fill it to the brim. And for the open beakers, we can always insert in cotton. you insert in cotton wool. So this is the cotton wool. Then for other instruments like the curvature plates and other glasswares, we wrap them into what we call autoclave bags, or you can use polystyrene. So this is the autoclave bags we use to cover other glasswares. Also for the surgical blades, the scissors we have used during surgery and other equipments are wrapped into what we call autoclave bags before we begin to autoclave. So after packing or loading the materials within the tray, we come and insert the tray. But before inserting the tray, we first fill the autoclave tank with water. So we come, when the outlet is closed, we fill it with water until water covers the heater or the gas. So after filling it, so we fill the autoclave with water. After filling the autoclave, we come and insert. So the first procedure is to fill the autoclave with water. Then you come and insert the loaded tray. So we come and insert the loaded tray into the autoclave with the materials to be sterilized. These are the materials which we already loaded to be sterilized. And while packing the materials, always make sure you leave space in between so that there is free circulation of steam, because we are using saturated steam, so we don't overpack. We must sure we leave spaces in between the autoclaved materials. And after packing them, always remember to add the control, such as the masking tape, the autoclave tape. Always on each material you put the autoclave tape as a control to see if the maximum sterilization was achieved. So we have filled the tank with water until it covers the heater. Then we come and load our tray, which is also perforated, with our materials to be autoclaved without forgetting to add the control as the autoclave tape. So after putting the tray in the autoclave, we have filled it with water, we have loaded it with the tray which has the materials to be autoclaved. After loading it, we come and cover. So we fill, then we load with the tray which contains materials to be autoclaved. After loading, we now come and cover tightly. Or we can use the word we secure the lid. And how do we secure the lid? By tightly closing the screws. The screws must be tightly closed so that we don't allow any steam to escape. So after securing the lid tightly, you open the air remover. So you come and open the air remover first. You open the air remover. So open the air remover or the air valve. After opening it, switch on from the power source. After switching on the autoclave, then press the on button. After pressing the on button, set the timer. And the holding time is 15 minutes. But depending on the materials, it can even go up to 20 or up to 30, depending on the materials being autoclaved. So after timing, when the water starts boiling, it is going to produce steam. And this steam will drive away all the air which was trapped in the autoclave via the air remover valve. That's why we first leave the air remover open to allow the steam to drive out to the air. Because if air remains in the autoclave, it will interfere with the steam and we cannot achieve maximum sterilization. So we first open the air valve remover so that the steam which is forming from water boiling drives out the air. And how do we know that all the air has been removed? You can connect a tube and you put it in air, you put it in water to see if there is air bubbles. If there is still air bubbles coming out, it means the air is still present. But if we see no more bubbling when we collect the tube from the valve to water and there is no bubbling, it means all the air has been removed. And when we ensure that all air has been removed, we come and close the air remover again. We close, and after closing, then our autoclave will begin heating up. That is, temperature will start to rise, and also the pressure, and it is seen on the pressure gauge, whereby the pressure gauge gives all the pressure and the temperature. So it can read the pressure at the same time the temperature. That is the pressure gauge which we are using. So you can check there. And whenever during sterilization, after switching on and it is heating and it has started sterilizing, during holding time, whenever the pressure goes beyond 15 pounds per square inch and the time goes beyond at 121 degrees centigrade, there is what we call the safety valve. The safety valve opens and allows steam to escape until it comes back to 15 pounds per square inch at 121. When it comes back, the air valve closes and does not allow steam to come out. So after we have achieved our maximum sterilization, after 15 minutes, the autoclave will make an alarm and you will begin the cooling phase, whereby when the alarm has gone and showing you that the holding time is done and sterilization has been achieved, what we do, we come and switch off the autoclave. After switching off the autoclave, we can even switch off from the power source and we leave it to cool down. And how do we leave it to cool down? Is by even opening the air valve or the safety valve so that the steam escapes until we make sure temperature has gone below 100 degrees centigrade. And when it has gone below 100 degrees centigrade, we can even open the air remover valve to make sure all the steam has been removed, and carefully we can unscrew the nuts and we open our autoclave and we can unload our sterilized material. And after removing the sterilized materials, the first thing you do is to check on the control of the autoclave tape. What are we checking? We are seeing if the autoclave tape, if this was the autoclave tape on the materials, it should develop black diagonal lines on each material being autoclaved. Because remember, you attach the tape on each material in the tray. So you check for complete sterilization by looking for the black diagonal lines in the control tape. And when you find them there, it means all materials have been sterilized by moist heat, or what we are calling saturated steam, which we said it sterilizes by coagulation and denaturing enzymes and structural proteins, hence killing all the microorganisms so that we can have sterile equipment to be used in the hospital. So this is what we can discuss with you in this session. What are the factors that affect maximum sterilization by autoclave? One is time, another one is pressure, another one is the air. Air, if you have not allowed air to be removed by forgetting to open the air remover valve, it can affect sterilization, also the time you use, and also the pressure at which it is being maintained. Those three factors affect the sterilization time. And another thing which affects is failure to use controls and also overpacking materials in a tray without leaving spaces where steam can reach on each article being sterilized. That is also very important to note when you are autoclaving materials. Always leave enough. And please always check the water level. We said the water level should be above the heater when you're using electric autoclave. It is also very important to always check the level of the water in the autoclave. Thank you so much for learning with us. Hope you have gained a lot. Please remember to like the video and share to many others that they may continue to learn with us. Thank you so much.